Okay, we have well over 100 people join, which is great. So yeah, thank you for everybody for joining us for this webinar, Cell-Based Screening 101. Hopefully it'll be useful and you'll learn something about cell-based screening and how we use it in drug discovery. Next slide, please, Alicia. Just waiting for that. Yes, brilliant. So yeah, my name's James Robinson. I'm a director at AstraZeneca. I run a team, an in vitro pharmacology screening team within the Discovery Sciences department. For this webinar, I'm joined by a few other colleagues from inside and outside AstraZeneca, and I'm just going to ask them quickly to introduce themselves so you know who they are when they start talking later. Maybe Alicia, you go first.
Hi everyone, I'm Alicia. So I work as a senior scientist here at DomainX and I work within assay biology. Um, and I also sit within the LRIG work group, and that is the early careers work group. So me and my group, we're the ones that put on various different events and different things for early careers people.
Thank you, Alicia. Mercedes.
Hello.
Yes.
Hi, my name is Mercedes. I am a director in AstraZeneca. I do run the high throughput cell screening group, and I am a cell biologist by training. Nice to meet you.
Thank you. Jesse.
Hello, everyone. My name is Jesse Peterson. I'm a group leader here at DomainX within the assay biology department, and I specialize in cell-based screenings. And I've been doing this thing for, I don't want to say how many years, but at least 10. Put it that way. But really nice to meet you all.
Thanks, Jesse. Madhu.
Everyone. I'm Madhu. I'm the Chief Scientific Officer for Inspero, and I'm with Jesse. I'm not going to tell you how long I've been doing this, but I'm trained as a molecular oncologist and assay biologist, and I'm excited to be here.
Thanks, Madhu. And then finally, Carl Johan.
Hi everyone, I'm Carl-Johan Likovius and I work at AstraZeneca here in Gothenburg in Sweden. And I lead the therapeutic oligonucleotide assay development team here within Discovery Sciences. Nice to meet you.
Thank you, Carl Johan, and thanks everybody. So these are all the experts you're going to be hearing from today. If you go to the next slide, please, Alicia. So this is a quick overview of what we're covering. So we're going to be talking about what is cell-based screening and how it's used in drug discovery. You'll hear about various things we measure in cell-based assays and the different types of cell models that we use in cell-based assays. We have time for a panel discussion at the end, but feel free to ask questions as we go along as well. But please do use the Q&A function in Zoom rather than the chat function. That enables us to keep on top of it a bit better, and we'll keep an eye out for any questions as we go along. But as I said, we've got the panel discussion at the end, so we can mop things up there. But yeah, please use the Q&A function for that. Next slide, please, Alicia. So yeah, what is a cell-based assay? Well, we're able to grow various different types of cells in the lab, and a cell-based assay is basically any time we treat these cell cultures with either compounds or maybe a gene knockout, those kind of perturbations, and then measure some kind of functional event in those cells. That is what a cell-based assay is, and we do that routinely in drug discovery. Cell-based assays are particularly important, I guess, because they occupy a bit of a sweet spot between where they offer, more physiological relevance than, biophysical or biochemical assays that use purified proteins, but they also offer more kind of scalability and ease of use than animal studies. So cell-based assays do occupy a really important kind of sweet spot there in drug discovery. Next slide, please. So this is a schematic of the kind of whole drug discovery process. Now, you guys are probably aware that this can be quite a long a painful process. Can be over 10 years sometimes to get from um, a sort of project inception through to regulatory approval for a drug. Cell-based assays would normally sit in the early discovery preclinical phase, so those blue boxes that you can see there. I think if you do the build, there's a circle that goes around that. Then but I should say that increasingly you'll see cell-based assays being used to inform clinical, more advanced types of cell-based assays anyway, being used to inform clinical studies as well, and more cell-based data going into IND filing. But traditionally, cell-based assays are used in this preclinical early discovery phases. And if you do the build, Alicia, it breaks that up. So this preclinical phase can be broken up into, as you can see at the bottom of the slide there, target identification, hit to lead, lead optimization, and then candidate drug shortlisting. So cell-based assays are important for all of this, and they're particularly important at the target identification, target validation phase, where before investing all of this time and effort into a new drug target, we want to demonstrate using, cell-based models of disease that a potential target of interest really does have disease linkage, really does have a role in the disease phenotype, and we use various different cell-based models for that. So that's really important right at the start there. As we move into the actual hit identification, so the kind of high throughput compound screening normally, depending on the modality, but that may or may not use, it can use cell-based assays, as Mercedes will talk about later, but it can equally use biophysical or biochemical assays at that point. But then when we move into hit to lead and lead optimization, cell-based assays become really critical for all projects there in terms of making sure we're driving towards the right molecules based on having a functional effect in cell-based models. And then in that kind of preclinical phase, you'll also find cell-based assays being used more and more in place of in vivo animal methods for looking at efficacy and safety in that candidate shortlisting phase. So cell-based assays getting more and more important and used all across this preclinical pipeline that you can see here. Next slide, please, Alicia. And then in terms of the types of models that the types of cell models that we use, maybe this is obvious, but if we are using cell-based assays for primary high throughput hit identification type screens, where we might be screening thousands or even millions of compounds, unsurprisingly we would look to use the simpler, cheaper, higher throughput types of cell assays there. As we kind of go through the drug discovery process and triage down our compound numbers, drive towards our favorite compounds, then we might move into the sort of lower throughput, but more advanced, more complex cell-based models where we can really check that we're having the phenotype that we need in our cell-based assays before we go into, animal or even clinical studies. And I think there's a bit more on that on the next slide. So when I talk about the simpler, more scalable cell reagents, I'm talking more about the right-hand side of this table, the stable cell lines. So these are cell lines that have been immortalized in some way so that they basically will grow indefinitely in the lab. This obviously makes them really easy to use, and they are very routinely used. However, this kind of immortalization process and sort of multiple rounds of culturing in the lab can cause some drift, and these immortalized cell lines might not be the perfect or truly reflect the biology of the source tissue. But they are very convenient to use, so they're very routinely used. And HeLa cells are an example here. And the picture you can see in the top right, that's Henrietta Lacks, that the HeLa cells are named after. And she's the lady who, it was her biopsy back in the 50s, I think, where the HeLa cells were originally derived from. And those cells are used all over the world today and have been for years and will be for years to come. So that's an interesting story where you can, I think there's been a movie made about that, which you can watch. And then on the left-hand side of the table here, you've got primary cell reagents. These are isolated directly from animal tissues or biopsies or healthy donors, but they are derived directly from tissues. Here we've got, maybe not so easy to work with. Maybe, they won't necessarily grow indefinitely. They might have quite fussy culture conditions, etc. But they are closer. Their biology more closely reflects the source tissue. And these primary cells are also going to be the kind of starter material for building up some of the more advanced models that we'll hear about later, like organoids, for example. Yeah, so that's the difference between your kind of mortalized cell lines and primary cells. Next slide, please, Alicia. Yes, and then whether we're talking about the primary cells or the immortalized cell lines, the simplest way to kind of culture them in the lab is in these 2D monolayers. Most cells do effectively quite like sticking to plastic in flasks or multi-well plates, or if they don't, we can encourage them to with other factors in the culture. This is the easiest way to work with these cells normally. It enables us to easily treat with compounds and get a kind of a uniform dosing with compounds. And there's lots of different endpoints we can measure in from these 2D monolayers. So, for example, we could do immunofluorescent imaging for levels of a protein of interest, for example, in response to a compound treatment. So this is kind of routine. Obviously, we can build these up into more complex 3D models that we will hear about later, but I think first we're going to hear a little bit more about the types of things we can measure in 2D first. And I will hand over to Jesse for that, I think, for an example there.
Thanks so much, James. So yes, I thought I'd give an example of sort of how we can use these relatively simple models to build up some complex disease scenarios in which we can ask very complex questions. And so this is what we call our scar in a jar assay. So it's an example, I think, of a simplified disease model that balances that need for modeling that complex physiology, but giving us enough tractability and scalability to do things at a moderate throughput that we would need in a drug screen. And so it can give us a clear readout of the effectiveness of a drug, even though it's not per se sort of a fully recapitulated physiological scenario. So if you're not familiar with fibrosis, it's a condition in which that normal normal wound response becomes dysregulated. So this can lead to uncontrolled tissue remodeling and scarring and a chronic inflammatory environment. And there is no cure for fibrosis. There are therapies that seek to slow its progression, and those are usually organ specific. It affects virtually every organ, and in the end can result in organ failure and death if left untreated. So mechanistically, what's happening is that these myofibroblasts, which are derived from fibroblasts and other sources in the tissue, are activated and start to deposit excessive extracellular matrix, or ECM. So what we can do with this assay, which starts with a very simple sort of cellular model, is to monitor the extracellular matrix deposition by looking at collagen or other ECM components. And we can look at myofibroblast activation by looking at smooth muscle actin in parallel. And so we get quite a few readouts at the same time. And you can see in the slide that it's an immunofluorescent readout. It's fairly moderate throughput, and it's we can scale it up for depending on the number of treatments we're trying to assess. So if you can just move to the next slide, Alicia. So what we're seeing here is an increase in the TGF beta. Hopefully you can see that bar on the top. So with increasing amounts of this pro-inflammatory signal, you have an increased collagen deposition. So these myofibroblasts are secreting more of the collagen. And if you move to the next slide, sorry, it's the next slide over. And now you can see what happens when we treat with an anti-inflammatory drug. So this drug specifically targets the TGF beta pathway, and you can see that that collagen deposition is decreased. And so we can get sort of a sense of the initial dose response with this kind of work. And it's an imaging assay as we're showing here, but the nice thing about it, and with many of these 2D assays that James described, is that we can multiplex them to monitor things like secreted markers of inflammation, or even look at gene expression from the cells themselves. So starting from a very simple tool and a very simple model of a degenerative disorder, we can really get quite a lot of information about the drug in an early stage. And with that, I'll hand it over to Mercedes.
Hello, thank you, Jesse. So yeah, I will speak a little bit about the use of cells in high throughput screening. As James introduced, after target validation, we do move in the process of hit identification. So we will have a target. We will want to identify chemical series and compounds. In this case, because I'm focusing on the small molecule, that they do hit this particular target. On the right-hand side, what you do have is a general workflow of how HTS look like. Normally, we do start with millions of compounds. This can be supported by either biochemical, biophysics, or cell screening, cell assays. After that, we would be moving into confirmation, then concentration response, which could be something similar to what Jesse just showed on his slide. And then we do 3/8 other assays. And the idea is that we will end up with between 0 or around 4 chemical series that then they will progress into the next stages. The more important part on the HTS process that involve cells is actually to balance the quality of the assay, which is our primary focus, with the time, because we need to screen millions of compounds, and this is something that normally takes around 1 month. And then the cost. So in order to balance of this, what we need to make sure when we do pick the assays and the models is to make sure that they are disease relevant, that they are simple enough as for us to be able to make conclusions and to make decisions, yes, no, at this stage. The assay format normally dictates also the size of the screen, because the more complex, the more complicated it is to put in automation, which we'll see in the next slide, and also probably it's going to be more expensive. So as a rule, what we do try is to balance the complexity, and the more complex, the less compounds, and the longer the duration. So normally our motto is as simple as possible, as complex as necessary. And this is how we are using cell screens for HTS. And in order to generate this high amount of data in this minimal amount of time, we do rely on robotics, liquid handle automation. And on the next slide, you will be able to see a short video of how our automation platform looks like. So what you can see here, maybe you need to press play a couple of times because it's going to end, and then what we do have is a platform where there is a central robotic arm that communicates with all the other cards, and also each of the cards, they do have their own robotic card as well, robotic arm as well. And what you can see here actually is the robots working, moving plates around. Normally we work in 1536 well plates. And yeah, everything is orchestrated in automation. So that is also another of the things that we need to have into consideration when we do choose the type of assay that we will be running. And with this, Johan will take over. Carl Johan, yes, thank you.
Yes, next slide, please. So one assay that you might have heard of is the cell painting assay. That's a cell-based one, which was developed at the Broad Institute in the US. And instead of using a targeted approach where you're doing a staining for a particular protein, for example, in cells, This is a more unbiased approach where we look at the morphological features of cells rather than particular proteins. You can advance to the next, please. So how it works is that we plate the cells and then use general fluorescent dyes to stain some of the major organelles of the cells. And you can see the list there. And then we use automated high-resolution imaging to capture these different stains. And together, from those images, you can do image analysis to extract the features. And that can give you sort of a fingerprint of the phenotype of the cells. And if you treat the cells with different compounds, for example, you can look at how they cluster. So you can see some images there showing clustering of different compounds. So this way you can compare with the compounds that have a known phenotype and a known effect. So in a toxicity assay, for example, you might compare with compounds that you know will induce liver toxicity in humans. And then you can check your compounds to see, do the phenotypes of my cells we treated with my compound, do they look more like the liver toxic compounds or more towards the liver safe compounds? And you can do similar things looking at the mode of action of compounds, for example, to try to decipher information about the compounds. Next slide, please. So James gave a bit of background on this, the use of more complex cell models, such as induced pluripotent stem cells and primary cells coming from patients or from animals. So this is used where we want to have a biological system that is more closely resembling the in vivo situation. And there we use these kinds of cells. And that can be used either for validating, to look at the compound effects, either during very small screens, so screening small sets of compounds, or used later down the screening process after you've run the high throughput screens in a more simple model, for example. So iPS cells, they are a more scalable version than the primary cells. They are derived from other types of cells, and then they are differentiated into the cell type that you're interested in. And that can be used where primary cells aren't available, for example, neurons or cardiomyocytes. These types of cells are much more challenging to work with than the traditional immortalized cell lines. They're also quite expensive. And with the primary cells, there's limited opportunity to expand them. They also often use proprietary cell media, which can be complicated because you don't know what's in the cell media. And they are also often resulting in less robust assays, which can be challenging. With the primary cells, you also have the added challenge that cells from different donors might behave in different ways. And that can be challenging for an assay where you want robustness. But it also provides opportunities to see, for example, differences in the human population. And you might get cells from patients, and you can compare them to healthy controls and so on. So there's a lot of benefits with using these cells as well. And ways to getting around this donor-to-donor variability might be to use pools of different donors together, for example. Yeah, next slide, please. And I'm handing over to Madhu.
Thank you, Carl. So a famous mathematician George Box once said, all models are wrong and some models are useful. And over the last 17 years screening, I cannot agree more. You've heard over the last 25 minutes, about different models and how you build in complexity, how you build in throughput for different models when you're answering different questions with different phenotypes. But I think it all comes down to really knowing what your biological question is and what assay really answers what particular question. And I think we're all, we came through that phase where we went from 96 to 384 to 1536. And then now we're coming off of that ledge back into 384 when we build in the complexity. Because, high throughput means different things to different people. And we're at a stage now where I think to answer any biological question, especially when it comes to disease pathologies, you may need more than one particular assay or one particular model to answer that question. So whether you're in 2D or 2.5D here, when you have iPSC-derived models or stem cell-derived models to answer target identification or target validation questions that you have early in discovery, or whether you go into spheroid or organoid models as you answer more sort of penetration questions, especially if it comes to oncology. Then you have bioprinted tissues, which are sort of much more further up the line, and then you have organ on a chip tissues. As you see, are sort of on the bottom layer over there, as you increase in physiological complexity, you do decrease in sort of throughput compatibility. And as we go on into sort of increasing throughput, you'll see that throughput really is less about biology and it's a lot about sort of combining physics into it as well. So that's something you really have to keep in mind when you start doing cell-based screening. It's not just about the biology, but it's about the physics as well. Next slide, please, Alicia. So this is sort of going, emphasizing a little bit more about the biology. How do you get deeper into the complexity, and what does it really mean in terms of this in vitro to in vivo translation, especially when it comes to the regulatory aspect, right? When we've all a lot of us, at least a lot of us, are doing this in vitro to in vivo translation or going into these more physiologically relevant models from the perspective of drug development. And there is this huge gap between what we're doing in the lab and what the regulatory authorities really want to see. And their definition of physiological relevance and clinical translatability of these 3D microphysiological models is a little different from what we see in the discovery world. So in terms of what we need to be able to provide for that in vitro to in vivo extrapolation in the cell-based screening world, this is sort of the emphasis or the sort of the main aspects of the spectrum from 2D to sort of model multi-organ physiology that needs to that we need to be able to provide from 2D cultures through in vivo physiology. So when it comes to 2D cultures, the reproducibility that we see have to be in cells, in the cell culture media, and in ECM proteins. And it may seem trivial, but the reproducibility and the robustness in these simple components is something that is not necessarily emphasized from lab to lab to lab. But if this is not given enough attention, early on in discovery, it's extremely difficult to sort of like get it through the more complicated models down the line. When it comes to 3D spheroids and organoids, and I'll get into the difference between these two in the next two slides. You're dealing with multiple cell types, you're dealing with multiple cell interactions, you're dealing with extracellular remodeling. There are multiple different interactions that have to be modeled really from a disease phenotype. And again, here, there is this context of use that has to be really defined for each disease pathophysiology. And if it isn't done correctly, again, the model may be beautiful, but it may not really answer the biological question. then you sort of get to the realm of microphysiological systems where you introduce this concept of vascular flow, where you now have that three-dimensional, geometry, but then you sort of add this flow and sort of shear force component, and again, it really changes. It changes the way that you you look at the pharmacokinetics and the pharmacodynamics of drug in a system, and it gets much closer to the in vivo biology. But again, now it is a much more complicated system and has very, very low throughput. Next slide, please, Alicia. So how can you really model this in high throughput screening? So this is an example of what you would see in to answer one specific biological question when you're going from very high throughput to low throughput in the same cellular system. So this was a screen that we did for it was high throughput pancreatic cell assay. We did a 1536 well assay in 2D, and I think we screened 100,000 compounds. And as you can see on the left-hand side, that these were 2D cell lines. This was a 2D cell assay where we screened 100,000 cell lines. When we took it into 3D, this was actually in a 1536 well plate, and you can see multiple different spheroids per well. And you can also see that it is not that reproducible because the spheroids are sort of different sizes. So the information that you're getting in that middle component with the 3D spheroid models is now very different from what you're getting on the left-hand side, in the 2D model. And that's why you're actually screening in 3D now. So you're not going to get information on a per spheroid basis. What you're going to get in this middle panel in terms of information, in terms of the hits that you get, is what is different between the 3D spheroid and the 2D spheroid, the 2D monolayer model with respect to the proteins that allow for adhesion to the sort of surface layer, and the ECM proteins and the sort of the cell adhesion proteins. Then when you go to the 3D organoid layer, that's when you have one organoid per well. You can't really get this in a 1536 format. You sort of get it in a 384 well format, and that's when you get much closer to really mimicking what's going on in a particular primary tumor, with all the zonation, with the zone of quiescence, with the zone of proliferation, and now you're getting a completely different set of hits. And the importance really of doing this entire spectrum is to understand the different biologies that you get with the exact same cell line for the spectrum of biological questions that you're asking for that particular tumor biology. Next slide, please, Alicia. And this is sort of a representation of that last organoid model that I was talking about. So when you think about tumor models, on the left-hand side, as you can see, there are these different gradients. I'm sorry, there are these different zones that you're going to see that represent these gradients that you see on the right-hand side. So in the middle, you have this necrotic zone, if you allow them to grow long enough, which we typically do because you want them to mimic what you will see in a primary tumor model. Then you do have a proliferating zone and you have a quiescent zone. And the quiescent zone is something that is extremely important, but very often not modeled in high throughput screening assays, because what we target are the necrotic zones and the proliferating zones with most of the chemotherapeutics. But in my experience, the quiescent zone is probably the most dangerous zone, because these are the cells that have acquired some sort of resistance to the chemotherapeutic or the targeted therapeutic, and they're the ones that really go into this quiescence or the sleeping phase, really, and then are able to sort of get through it get through it, they don't die, and then they sort of repopulate the tumor after a period of time. So it's this quiescent zone that we really have to focus on, and where you find the most sort of cancer stem-like cells, and you have to eradicate them. Next slide, please, Alicia. And last but not least, I think we've had a lot of excitement, I suppose one could say, over the last 12 months with the FDA and the NIH really talking about these non-animal non-animal methods for high throughput screening. While there has been a lot of excitement, I would say that this is not necessarily something that's new. It has been something that high throughput screening communities have been doing over the last 10 years, 12 years, both in the efficacy realms as well as in toxicology. What I do think is exciting is that there's a whole lot of what I do think is exciting is that there is a whole lot of enthusiasm now, more on the regulatory side, for people to now accept the data that people generate into making more regulatory decisions versus just sort of keeping it in the discovery realm. And with that, I think this was my last slide, and I hand it back to you, Alicia.
Yes. Oh, we have just this one slide if you wanted to go through it. If not,
that's right. This was my last slide. Thanks, Alicia. And this, I think, this slide I had in here to show you sort of the changing face of regulations from 2022 through to 2026. Actually, the last FDA announcement was in 2025, but we've seen actually the beginning of 2026, though there was no announcement that was made. The NIH SAM Institute that announced this $87 million initiative for the standardization of organoids. I think they finally announced that they would release the money for the institute early in 2026. The importance really of showing this this table over here is to show you how the language has changed over the years, because with the FDA Modernization Acts 2.0 and 3.0, it was always a suggestion with respect to 3D assays. They removed mandatory animal testing, but they never really pressured anyone into developing 3D models. So it was always a very light suggestion. You know, if you wanted to develop these models, if you wanted to submit this data, you were welcome to do so. But I think with the regulatory announcement and the FDA roadmap in April 2025, there was really a push to now start submitting this data, and there was a strategic decision made to eliminate animal testing by, in the next 3 to 5 years. So I think, going back to my earlier point on the previous slide, even though this is nothing new and we've been doing this for the last 12 years, there is now a timeline to get rid of animal testing, which I believe is a good thing.
lovely. Thank you very much. Thank you all also for the other panelists and taking us through their slides. Just while we've got lots of questions coming in, and we really appreciate that, just before we get to them, and while you're typing them and getting them into us, I'm just going to quickly go through, if you are heading to Gothenburg, that we do have some ECP specific things going on. So each morning there will be a breakfast, so you can meet. I will be there at 8:15, as will some other members of ELRIG, and we'll be there so we can have a catch up and you can meet other people before we go into the conference. We of course have the flash post presentations, and these will be taking place in the auditorium, there's also the networking at 5 o'clock on the Wednesday. And some tips is just to get out there and to speak to people. If you're feeling a bit nervous, just get speaking. The vendors want to talk about their products and their services, and the presenters also want to speak about their posters as well. People want to speak to you as much as you want to speak to them. If you've enjoyed this webinar, we also do have some previous webinars as well. And we also have one coming up where the registration is now live. So please sign up to this. And this is looking more into the careers beyond the lab and into the IP and the business strategies and those sorts of careers. So let's go through some of our questions. I know some have been answered as we've been going through. So let's find one. So we have one regarding cell painting assay. So for the 2D cell model, do you see the need for applying the same also to 3D models and the MPS systems as well?
I mean, that could be very interesting. The thing is that, and it has been tested actually, cell painting in 3D systems. The thing is that the image analysis in 3D is very, very complicated. And also acquisitioning of all the images in 3D, because you're acquiring a lot of different channels, and in 3D you're also doing at different Z heights. It's not an easy assay actually, but it could be very, very interesting, I think.
Lovely. When you mentioned there like a little bit of the challenges, so we had a previous question come in, and it was about the challenges and the limitations when you're trying to move from cell-based screening into clinically relevant outcomes. So Madhu, would you like to take this question? So how are we addressing these issues as they're happening?
Yep, happy to take that question. So I think it's a very relevant question. We've over time, I think we have had to segue from metabolic readouts like ATP and LDH. And I mean, not that we aren't doing it now, but we are segueing from readouts like ATP and LDH and moving more sort of into the biomarker realm with readouts like albumin, AST, ALT. And other sort of clinically relevant biomarkers. And by that, what I mean is it's not that ATP and LDH and other sort of screening readouts are not relevant. What is relevant, what is needed is to be able to connect the dots between those readouts and clinically relevant outcomes or clinically relevant biomarkers. And if you're able to sort of make that connection, then when you're doing the high throughput screening, when you're still screening in 1536, and you're going from sort of that hit to lead phase, you're still able to screen in that and then go into sort of a lower throughput and then screen sort of clinically relevant or functional biomarkers.
Lovely, thank you. We also had a previous question about whether if we can make 3D work and organoids more high throughput, will 2D cell culture still have a place in the lab, and can we still utilize that?
Yes, I think sorry, go ahead.
No,
Was
you
there
go ahead.
someone who said something?
Yeah, I was going to, but you go
No,
ahead. I
go
was going to
ahead,
give you
James.
a
No,
break.
no, go ahead.
I think it comes down to the point that Madhu was making earlier about it depends on the question we're asking. Um, there's definitely a place for simpler, more 2D, more scalable systems in the lab because, very early in drug discovery, we might just be asking the question of can we find any chemistry that actually modulates the function of that target? And as long as we've got a way of faithfully in a robust way monitoring the function of that target, we might as well do that in as simple a way as possible at that stage. If your question is, do I have a compound that will modulate a disease phenotype, or does this mechanism, at this target, if we modulate the function of this target in this way, does it have the desired disease phenotype? Or, in oncology, often it's would that combine well with, a current clinical standard of care or chemotherapy or something like that. Those types of questions are where you need the more advanced models. But if you're asking the question of, can I find a compound, a very early weak at that stage compound that a chemist would be interested in working on and optimizing, by screening lots of different compounds to try and find that, then as long as you've got a way that you truly believe you're in a robust way monitoring the function of your target, you might as well do that in as simple a way as possible. So it definitely depends on the question. And this, we'll use all different types of models depending on the question.
Lovely. Thank you very much. So we've got a couple of questions about high throughput screening. So someone here is interested in doing a high throughput screening, but currently doesn't have a lab that's got access to the automation. So they're wondering if computational screening and AI could assist with this aspect.
Yeah, I can reply to that. I think that is also related with another question that we had before about what is the current state of the AI and computational. So I think that the short answer is that it will depend a lot on what the target is. So if there is something that we do have good knowledge about the target, the structure, etc., we can do a lot of computational and AI upfront. That's what we do call the knowledge-based screening. With that, we can filter out and we don't need to then screen millions of compounds. Then that can reduce the list to maybe just a couple of thousand or sometimes hundreds, depending also on what is also our assays and our systems in the lab. If the target is very novel and considered undruggable, this is something that we are starting to work with. And for those, we will need to first generate a good amount of data before then being able to do models and predictions. So yeah, it mainly will depend on the target and what we have in place in the lab to query those hypotheses.
You mentioned there about the design and shaping things for your target. So someone also asked previously about the designing as we're shifting. Well, high throughput is a lot about a numbers game, and as we are shifting into this AI era, if the bottleneck is shifting from the raw throughput to the richness of data, and if we're moving beyond the yes and no hits, how are we going to redesign screening to prioritize capturing this high dimensional data without degrading operational efficiency?
Wow, that's
quite a long question.
That's an impressive question, whoever asked that.
Yeah, I would say that maybe we are 5 years away from having something really, really good in place. We are starting to do more phenotypic screenings. We are starting to use more cell paint, but the complexity is also not only on the assay or on the limitations about the source of your cells, if you can use this many or just an spheroid. The complexity is that the more data you do have, your matrix is also expanding a lot. And then you do have maybe 20, 30 different characteristics associated with your compound that you will need to decide if you do progress further or not. So all that we do need a quantitative biology, bioinformaticians, people that they can do some also of the genetic overlay into what the different samples are expressing. And it's a whole infrastructure. It's not only about having automation or having the feasibility of doing an assay. It's also where then computational chemistry, biology analysis, and all that process the data that then eventually it will be possible to see on an interface where a chemist is going to be able to get a score or something that summarizes all that data to then make the next stage and the next conclusion.
Yeah, I think there are some endpoints where we already have a lot of data. So like safety endpoints and phys-chem type endpoints, the models might already be quite good. And we may be already moving towards more of a predict first mentality there. But as Mercedes was saying, with like unprecedented targets, we still need to generate a lot of data before we have, powerful models. There might be a future in 5 years' time, as Mercedes was saying, where we have these foundational data sets and we can do a lot more predict first across the board. But at the moment, there's still a lot of data generation required, and the data quality and the verification of data is what's critical at this point in time, so that it's reusable and searchable in the future.
You mentioned there a lot about moving from, or you're getting these large data sets, and then as we saw, as you're moving through the cascade, and then you're moving along through the early phases. Someone's asking if when you're moving from a hit to lead, is it common to see loss of promising hit compounds because of poor cellular accumulation within the target cell? Jesse, did you want to take this one?
So I think Mercedes is typing an answer at the moment.
Yeah, but please go ahead, Jesse.
Yeah, but I was just wanted to sort of comment on the fact that at this point, this is why you want sort of diversity, why you are testing quite a few compounds at this stage. So you do see things like drug not getting into cells, not reaching a concentration that's effective in the cell. But again, you don't put all your eggs in one basket at the lead phase.
Yeah. So this is obviously something we are all very well aware of as working in early stage discovery, is we're screening lots of compounds and not a lot of them are getting through. So someone recently saw a stat about a large percentage of compounds failing in clinical phases, and they're wondering if they think this is an issue with the models we use, or is it other reasons?
I would encourage people to read the AstraZeneca 5Rs paper. There's been 2 of those papers now, because it talks about the reason for failure. Because it used to be that we would fail mostly for safety reasons, but now we're actually quite good at preclinical toxicity. So we fail more now for efficacy reasons, and or at least being able to achieve doses that drive efficacy. So I think that does open the question of, are we good enough at predicting efficacy, or are we good enough at choosing the right targets that will be efficacious in the disease setting? And that's where there is a drive towards these more translatable cell models, but it's a massive challenge. I think it's probably the biggest challenge is demonstrating translatability of preclinical models where maybe we don't have the clinical data for that mechanism or that target yet. So then we have to build up, omics data sets and things that convince us that we're working with the right models, do pharmacological validation where we can. But it is a big challenge to convince ourselves, where we don't have the clinical data for a lot of these mechanisms, that the models are translatable. But yeah, I think if you read those 5Rs paper, it's interesting to see how the reasons for clinical failure have changed over the years. Don't know if anyone else wants to add anything to that.
I think it also highlights the needs for the more advanced cell models, where we can use patient sample cells, for example. I mean, if we go for a hex cell and then go into and testing it into mouse models, we might find things that work in vivo in mouse models, but then taking that to the clinic, it might not work in humans because we function in different ways.
Yeah. Just looking for some more. So a lot of people they tend to be wondering about what is our, the biggest challenges that we face. So I think we can split this up into maybe the biggest challenges we face within high throughput screening. And then when we're trying to, let's go for the high throughput screening first. Mercedes, would you like to answer that one?
In my case, I would say that within the last couple of years, the bigger challenge it has been all the undruggable portfolio, where it is very difficult to choose what is the right assay, and also because we don't have a really good understanding on what is the chemical equity that it could hit, then we need to do as many compounds as possible, and that limits also the type of assay that we want to do, because probably we would like to do for those something a little bit more complicated, like going into imaging stainings or 3D But balancing those two things at this stage and balancing it with the cost and the throughput, I would say that is our current bigger challenge.
Nice. And then when we're actually, Carl Johan, did you want to take, when we're actually selecting or designing a particular cell-based assay within early drug discovery, what are some of the key factors that we have to balance and consider when we're trying to balance the physiological relevance with then the scalability? So is there any particular challenges that we face?
Yeah, I mean, of course, it would be nice if we could run high throughput screening in patient samples in 3D models representing really the biology. But the thing is that it would become extremely expensive. And also the readouts in these more advanced models tend to be longer. So if you run imaging, for example, it takes a fair bit amount to do that in a 3D model in particular. So that's why the high throughput screens are usually done with a more simple system, and then further down the line you can go for more advanced systems. Also, if you're talking about primary cells, there's the availability of the cells isn't huge. Because obviously these come from people, so you can't get an unlimited amount of cells from them. Often it can be quite limited. Even with iPS cells, where you can expand them, they are extremely expensive. So that prohibits a lot of these larger screens. But I think the use of predictive models and AI could sort of bridge some of that. So if you use predictive models to identify smaller sets of compounds, you might be able to go straight into the more relevant cell models. I think that's quite an exciting opportunity.
I can also jump in on Carl's points there that the molecular tools available to us start to sort of diminish as the model gets more complex. So at the moment, we have tools that we can use in 2D cells that are very difficult to translate or even just to interpret in a 3D cell model, but that's changing, and you can see a lot of these tools being adapted for that. So it's exciting times.
Are there any? Sorry, no.
Sorry.
Matu?
No, I'll just add one more thing to the 3D model space. I think robustness is key, robustness and reproducibility, especially if we, as we move into testing patient models, you're going to see the interpatient variability because that's what you want to model. We want to model that patient heterogeneity that we see in a clinical trial population in discovery, right? We want to have that in the preclinical space. But if you don't have the assay robustness in your preclinical assay, then you don't know if the variability is coming because of that interpatient variability or because of your assay variability. So I think that's something key to keep in mind, as you get sort of into these more complex models.
Lovely, thank you. I'm just aware on the time, and I just thought we could end on, is there any future advancements or any technologies or anything that we're excited, looking forward into the future that will be coming up? I don't know who wants to go first, or if anyone has an answer.
Probably the answer is AI, but I'm not going to say that because no, I think I guess I'm most excited about the momentum behind using preclinical in vitro models to better predict clinical outcomes, and that actually being accepted by the regulatory authorities, as Madhu's been already explained very well. That's a massive opportunity for us that work in this space. It's an opportunity to really accelerate drug discovery in a more kind of ethical way as well. So that's really exciting for me, I think.
Lovely. Yeah, Jesse, please go.
Jump in with one. So yeah, just my own sort of pet interest is the new generation of biosensors that are starting to emerge. So sort of ways that we can monitor biology in real time in live cells, and increasing our sensitivity by using things like novel proteins, de novo proteins, and all the work coming out from there.
Lovely. Thank you. That's really interesting to see. It's been really nice to recap on what our current work is and what we have go through, and then the challenges we face, but then also where science might be heading in the future. So I'm aware of the time. So thank you everyone for sending in your questions. We will do our best to get some answers to you. If we haven't been able to answer them already, we will try to. I'd just like to thank all the panelists for joining us today. It's been so nice to hear from you and to have your expert opinions. And thank you also everyone for joining us online. Does anyone else on the panel have anything else to say? If not, then thank you very much.
Thank you.
Thank you.
Thank you.
Thank you.
Bye.
Bye-bye.